Good morning, afternoon, or evening, depending on where you're coming from. So it's really exciting that we've seen folks that have joined us from all over the globe today, and hope you've enjoyed the sessions we've had so far. The session that we're going to have now is kind of a hybrid session. So this session is a combination of using a recording that has already been pre-recorded, but then we are super fortunate because we have Dr. Anton McCaffrey who has joined us, and he's going to be able to answer your questions. So you'll have a recording of both from Jessica and from Anton, and I'll tell you a little bit about who they are, and then afterwards, we'll be able to get your questions live. So just to tell you a little bit about what the session is the basics of RNA and RNA vaccines, and we know that that's been a huge topic and has been for the last couple months and has been for some people for several years. And so one of the questions is, what are the basics and where am I going to get these vaccines? And so we're very fortunate today to have Dr. Anton McCaffrey, who is the Senior Director of Emerging Science and Innovation at TriLink Biotechnologies. He has joined us today, and he has over 25 years of experience designing nucleic acid-based therapeutics. And so he's going to be able to bring quite a bit of experience to this conversation. The second partner, after he presents, then we'll have a recording, stays on, same session, nothing changes. And then we'll have Jessica Madigan. She's the Director of Business Development at TriLink Biotechnologies, and she's played a pivotal role in the pioneering of contract messenger RNA manufacturing. And she has served as a manufacturing support lead for groundbreaking programs in mRNA therapeutics. And of course, that's really kind of the hot topic of the day right now. And that is that whole idea of how do you get it to manufacturing? So you really don't need any more of me. What we're going to do now is going to go straight to the video. And then, like I said, immediately following the video, Anton will be joining us to answer your questions. So we're going to turn it over to Natalie, and she's going to run the video for us. So just give us one quick second to get everything started. I just got it perfect. And we are ready to go to video.
Hi, my name is Anton McCaffrey, and I'm Senior Director of Emerging Science and Innovation at TriLink Biotechnologies. And today I'm going to tell you a little bit about the basics of RNA and RNA vaccines. So first, just a little bit about TriLink. So we're a contract manufacturer, and we were the original contract manufacturer and most experienced contract manufacturer for messenger RNA. In recent days, we're adding a new plasmid capability for GMP-grade plasmids. We offer a variety of different analytical services, and we have 8 clean room GMP suites for manufacturing of messenger RNAs, oligonucleotides, small molecules, NTPs. We manufacture NTPs and do custom chemistry. We also have some proprietary capping technologies called CleanCap that I'll tell you about in a minute. And our core competency over the years has been highly modified oligonucleotides. And we have over 20 years experience with this particular platform. So as I mentioned, we're a contract manufacturer. We don't manufacture any of our own drugs. So you don't need to partner with us and have a partnership in order to manufacture. We're just a contract manufacturer. And we have a whole spectrum of customers. So some customers come to us and they have batch records and they simply want us to execute protocols that they've already established in a GMP setting. On the other side of the spectrum, we have people, for example, who might have come from the viral vector world, who know that they want to do messenger RNA, but don't know exactly how to go about doing that. And we can provide them with a complete solution to lead them to GMP pharmaceutical manufacturing. So, as I mentioned, we've been doing this for quite a long time. We started manufacturing mRNAs in 2011, and we initiated a build of a GMP facility in 2015. And just last year, we moved into a larger facility that I'll tell you about a little bit later on that has greatly expanded our GMP manufacturing capabilities. So what are common applications of messenger RNAs? Well, they're extensively used for genome editing, for example, zinc finger nucleases and talons, or CRISPR and Cas9. They can be used for gene replacement. They're quite popular in the field of immunobiology with CAR T-cell production or personalized cancer vaccines. They also are popular in infectious disease for mRNA vaccines that you've heard a lot about in the news lately, and for self-amplifying RNA vaccines or SAM vaccines, which I'll tell you about a little bit more later on. So as I mentioned or alluded to a little bit earlier, there are several types of mRNA vaccines. So they're unmodified RNAs. then usually you'd require a purification to remove double-stranded RNA that might not be desirable. There are modified mRNAs that are substituted with modified bases that substitute for uridine that increase expression. And then there's self-amplifying messenger RNAs, and these are unmodified so they don't interfere with RNA replication. So during your planning of your GMP process, you want to pay attention to a number of different things. So one is that the reagent quality and attributes should be appropriate for different stages of development. So you may need different grade materials for research grade or preclinical, and different materials later on for late stage clinical development and commercialization. And this can include transcription templates and enzymes, NTPs, capping reagents. And the GMP process is well defined to minimize the number of critical reagents and steps, and this is important, and I'll get back to that a little bit later. You want the immunogenicity to be appropriate for the target's application, and this can be modulated with capping, chemical modifications, and purification strategies, and I'll touch on each of these a little bit later. Qualification and release of the assay should be stage appropriate. And in your GMP manufacturing, environmental controls and documentation are again stage appropriate. So before I get into it too much, I need to let you know a little bit about how cells recognize foreign RNAs. So there are pattern recognition receptors in the cell that are there to recognize pattern recognition, are patterns that are present in pathogens. And these can be, for example, the cap, methylated ends, double-stranded RNA structures, that kind of thing. And so when we're designing an RNA therapeutic, we need to be aware of these sensors, and we need to take steps to appropriately interact with them. Now, it was shown by Katie Carrico and Drew Weissman that you could do modification of mRNAs with pseudouridine or N1-methylpseudouridine or 5-methoxyuridine in combination with uridine depletion. And this reduced binding to innate immune sensors in vitro, reduced toxicity, prolonged expression in cultured cells, and increased translation. And so chemical modifications may be desirable for mRNA vaccines, however, not for the self-amplifying RNA vaccines I'll talk about more in this talk. One of the other elements that's important for innate immune recognition is the cap structure. So when the cap structure has an OH at this first transcribed sugar, this structure is called cap 0, and this is rapidly inactivated in vivo by the interferon response. If you have a 2' methyl group at this first sugar position, then this structure is called cap 1, and this is found in most higher eukaryotic mRNAs, and it persists much longer in vivo than cap 0. And so basically, to put it simply, you want a cap 1 structure. Another thing to consider in your process is mRNA purification. So you can use silica membrane purification, such as an RNeasy, for small-scale purification, but it's not automatable or scalable. And at TriLink, we've developed two complementary chromatographic purification methods. One called LC isolation, which removes residual proteins, DNA, and NTPs, but it doesn't remove double-stranded RNA. However, as I'll get to in a minute, that can be advantageous for SAM vectors as an adjuvant. The other method is a reverse phase HPLC, and this decreases double-stranded RNA and enriches for full-length mRNA. However, longer sequences can be challenging to purify. All right, so self-amplifying RNAs, or SAM. There's an exciting new platform for vaccine development. These are frequently derived from alpha virus backbones, where the antigen replaces the structural protein, such as a capsid, such that the replicon cannot make an infectious virus. They replicate through a double-stranded RNA intermediate, and thus they stimulate innate immunity and enhance vaccination. Another advantage is that tens of thousands of copies are made from each original copy in transfected cells, and thus you require very low doses. And as I'll show you in a moment, new vaccines can be produced very rapidly in weeks or months in response to pandemics. So this is what a self-amplifying RNA cycle looks like. In general, these are packaged as a positive strand genome in a lipid nanoparticle. Once these enter the cell, the positive strand RNA genome is translated to produce the RNA-dependent RNA polymerase from the non-structural proteins. This replicates the double-stranded RNA into a negative-stranded intermediate. And for a time, there's a double-stranded intermediate there that's sensed by innate immune sensors such as PKR, MDA5, and RIG-I. And this enhances immunity because it provides a danger signal in combination with presentation of the antigen, which is expressed from the subgenomic promoter. And this gives very nice immune responses. However, there's a little bit of a conundrum with alpha viruses, and that is that alpha viruses have a very conserved 5' end starting in AU. However, T7 polymerase really strongly prefers to initiate with a G residue. And so what researchers have done is they generally append a G to the beginning so that they can in vitro transcribe these RNAs. However, the Frolov lab has shown that if you append sequences to the 5' end of an alpha virus and passage these through cells, that they rapidly lose those additional sequences and revert back to the 5' AU, suggesting that there's some kind of a selective pressure that wants the end of the RNA to start with AU. Now, people use enzymatic capping to cap these RNAs with G. However, these replicons are extremely long, 9 to 12 kbs, and the enzymatic capping enzymes are expensive at scale and inefficient on structured 5' ends, and thus require heating to disrupt the structure, which is shown here, and this can lead to degradation of the mRNA. So there's another alternative, which is to use an anti-reverse cap analog, which again requires starting with the G. However, these have very low transcription yields, low capping efficiency of about 70%, and they yield a cap zero structure, which is rapidly inactivated in vivo or in primary cells. At TriLink, to solve these problems, we developed a solution called CleanCap AU. And so that molecule is shown here and here. So instead of starting with an NTP or a cap analog, we're starting with a capped dimer here that allows us to start with A and U at the first two positions. Now, in terms of comparing it to the anti-reverse cap analog, we have no need to starve the reaction for G. We get about 2.5 times the yield that we get with the ARCA. And the capping is not influenced by the 5' structure. And we get a one-pot reaction with no need for a purification step that yields a very high 4 milligrams per mil. Also, we have CleanCap AG for applications that don't involve alpha viruses. And in order to monitor the capping step, we developed a capping assay where we basically cleave off the 5' end of an RNA, and then we analyze that fragment by LCMS. And here you can see 3 different capping reactions. On the left is ARCA, where you get about 70% capped and 30% 5' triphosphate. Enzymatic capping can be incomplete, and so here you see partially capped intermediates. And then on the right, you can see that with CleanCap, we get extremely high levels of capping. And so we think that CleanCap AU accelerates the vaccine process in a number of important ways. So first of all, it reduces the number of steps that are required and can be done in a single pot reaction, which shortens manufacturing times. It also importantly reduces the number of enzymes that need to be qualified by 2. And this can dramatically decrease your cost. It's highly reproducible and eliminates the variability seen in enzymatic capping. And compared to anti-reverse cap analog, we get about 3 times the yield of cap material. And so we're quite excited about this, and this has been rapidly adopted within the field. Here's just a case study, which we did with Imperial College of London, Robin Shattuck's group. And you can see that within a very short time from being contacted by the client, we were starting to produce research grade material, and then we shipped GMP grade material and finished within really a very, very, very short time. And so you can see that these timelines can be highly accelerated in a pandemic type response. Last year, just about exactly a year ago, we moved into a brand new purpose-built facility with 105 square feet of potential manufacturing space. And it was custom designed And we have 5 GMP suites with ISO class 7 and ISO class 8 suites and class 5 hoods. Recently, we added 2 more GMP suites for producing plasmid. And these are produced in class 8 clean rooms for GMP manufacturing. Each of these suites has 3 different rooms so that each operation, for example, fermentation, lysis, and purification happens in a separate room. All right? And then we use single-use consumables throughout the process to minimize any kind of risks. We have ISO class 5 fill-finish biosafety counts. And this facility should be coming online in Q1 of next year. As I mentioned, you want your processes to be stage appropriate. And at TriLink, we can guide you through this, whether it's making preclinical tox material, phase 1 material for your GMP manufacturing. We can help you with process scale up and phase 2 manufacturing for ex vivo applications, or we can do tech transfer to a third party CDMO. One of the advantages we think of bringing in this plasmid production in-house is that this is going to reduce the amount of time it takes for you to manufacture. If you look at the bottom timeline, in general, we're having to put in buffer periods such that the queue of the plasmid CDMO meshes with our mRNA queue. By bringing this process in-house, we're able to reduce the amount of time required by about 50 days. And so we're very excited to bring this new We also have a highly specialized quality system. So we're ISO 9001, 2015. We're compliant with ICH Q7 Section 19 guidelines. All of the mRNAs and in fact, all the molecules made at TriLink are fully traceable, whether they're research use or GMP. We're frequently audited, and we have a dedicated staff to support that. And we can provide material grades all the way from basic research to diagnostics, preclinical tox, and clinical materials. So we can also guide you as to what grade of materials you need for your different applications, whether that's RUO, GMP link, which is sort of in between, and GMP. And these have different requirements at each step. And later on, Jessica Madigan will be giving a presentation and can speak to this in more detail. So assay selection is also important. So we reference ICH Q6A, test procedures and acceptance criteria for new drug substances. And so we can do ID testing, strength, such concentration, purity, impurity testing, safety testing, process-related impurities. And so we offer a variety of tests to meet your requirements. And if we don't have a test that meets your requirements, we may be able to develop one for you. So this is a question we get a lot from customers is how do I set specifications? And a lot of people think that the manufacturer would set the specifications. However, really it's the client that sets the specifications and that's dependent on their application, the stage of development they are at, and existing technology that is available to meet certain specifications. And while you will be setting the specification, we can provide you with guidance on available assays and their limitations. And this process usually requires extensive communications between client, regulatory personnel, and TriLink. And so it's important to start that discussion between your quality group and our quality group early on. And one other thing I might mention is the amounts required for final release and retention assays in some cases can exceed the amount needed for the actual study if you're doing very small studies. And so that's something to keep in mind. So we have a variety of assay qualifications that we can do depending on what exactly you need. And this is just a table that shows you some of those. And the method should be qualified. And it's important to note that average qualification timelines are 6 to 12 weeks. And so you want to plan that into your time. Don't forget, you also going to need to be able to do a stability study. And again, we can guide you through that and provide you with a variety of different solutions for that. We offer a variety of assays, storage temperatures, time points to meet these requirements. And we're happy to discuss with your team exactly how to carry these out. And just before I finish up here, I just wanted to put in a plug for a webinar that we're doing on Thursday. This is with 2 of the founders of the field. So Caitlin Carrico and Drew Weissman. So when we opened our facility on the 20th of November in 2019, the city of San Diego designated that date to be mRNA Day in perpetuity. And so in celebration of the first anniversary of mRNA Day, we're going to have a two-part seminar with Dr. Carrico giving us a history of mRNA therapeutics and their development, and Drew Weissman telling us his opinions about what are the futures of messenger RNA therapeutics. And so we invite you to register for free at trilinkbiotech.com/events. And we hope to see you there next Thursday. And lastly, we have a team of GMP experts, one of which, Jessica Madigan, his bottom right corner here, will be speaking as well today. And they can guide you through this whole process. So when you come to us with a GMP project, you'll have a dedicated person who will be your point person to guide you through this process and connect you with the different teams at TriLink and our external vendors that you need to communicate with. And so I encourage you to contact them at sales@trilinkbiotech.com or gmpsales@trilinkbiotech.com. And again, Jessica Madigan is going to be available later after her talk for a chat. And so if you have specific questions about the GMP process, I encourage you to speak with her. And with that, I'll stop and take your questions.
Thanks so much for making some time for us to finish this presentation today. And I also just want to thank Anton for covering the important considerations for mRNA manufacturing prior to me jumping in. So for me, I'm going to take the next couple minutes to cover additional considerations for GMP manufacturing, including timelines, facilities, quality control, specifications, and stability. So what I'm showing here is pretty much what we consider kind of a hair on fire timeline. This timeline is an actual representation of what we were able to accomplish for the development of a SARS-CoV-2 vaccine that is currently in clinical trials. If you add up all the days here, it totals about 63 days. And so this is extremely accelerated. Average timelines for standard clinical development of an mRNA manufacturing is approximately 9 months. So you're able to basically leverage TriLink's platform manufacturing process for accelerating these clinical timelines. In the next slide here, I am showing our new manufacturing facility, which we moved into in 2020. This manufacturing facility is basically our commitment to growing with our clients through the manufacturing phases. We have about 105,000 square feet of facility, and within that facility there are 5 manufacturing suites for GMP manufacturing. So we have the ability to do parallel manufacturing of 5 different clinical programs at any given time. Here I'm showing the traditional stages of therapeutic development pathway. Research grade material is typically used for toxin preclinical production, and GMP material is used for phase 1. One option to condense timelines for a clinical program is to produce a single batch as tox and phase 1 under GMP conditions. This would be considered a bit of a risk to the program because you wouldn't have relevant animal data to know that the material will be effective. But it definitely will help you accelerate timelines. And so if you want to take that risk-based approach, TriLink is here to help assist with that. After successful phase 1, we would move to a scale-up process where we're going to be making sure that the manufacturing process is achieving the amount of material needed for later phase production. And once that process development has been completed, you would move again to GMP production for later phase materials. When selecting a CMO, it's important to choose one that can meet all of your quality needs throughout the different phases of your program. So TriLink has the ability to manufacture basic research, diagnostic, and preclinical and clinical materials. But everything that we manufacture here, even for your research grade syntheses, is fully traceable and documented under ISO 9001 conditions. And our GMP facility is in compliance with ICH Q7 Section 19. A lot of the times when considering a new therapeutic development, the question of what grade of material do you need comes up. And you need to consider what grade of material not only do you need to execute your clinical trial, but also the raw materials that enter that process. So if you read the guidance from regulatory agencies, it does state that GMP grade materials should be used to make GMP grade product. But oftentimes GMP grade materials can be cost prohibitive or timeline prohibitive depending on what the scope of your scale or your program is. So you will need to do kind of a risk-based approach, and we encourage our clients to interface early with regulatory agencies in pre-IND meetings in order to establish what quality of raw materials is going to be suitable for their program, especially with respect to enzymes and plasmid. So now after you've kind of developed your manufacturing process, you need to start considering which assays need to be used in order to release your product for phase 1. By referencing the guidances, you'll see that you'll need to have ID testing, strength testing, and impurity testing. So we're always encouraging our clients to choose a robust panel of assays. At TriLink, we offer a number of different options for purity, including fragment analyzer and bioanalyzer options and HPLC options. For safety testing, you're going to want to include endotoxin and bioburden. And then you're going to want to look at process-related impurities, such as residual DNA and residual protein, and potentially residual solvents, depending on what type of manufacturing method you're using. After you've selected the assays, you're going to want to set specifications. The important thing here is to make sure that your specifications are achievable, and they're going to be dependent on the stage of development. So we encourage our clients in early phase to make sure that the specifications are as broad as possible so that as you advance through later phases, you can begin to tighten those specifications. And ultimately, it is the customer's decision to design what specifications are going to be met for likelihood of regulatory acceptance, but we're going to guide you on what's achievable. So for example, for a self-replicating messenger RNA that's going to be greater than 10,000 bases, setting a purity of greater than 90% by CE is not going to be achievable. So you need to make sure that you have efficacy data that demonstrates what purity is achievable and set the specs accordingly. And then it's important to note that final release and retention for GMP is actually probably going to exceed the amount you need for your study, especially for phase 1. So you need to consider that when setting manufacturing scales. After you've set specifications and decided which assays you're using, then you must make sure that the assays are fit for use. Methods should be qualified, and so you're going to want to look at a range of options, including accuracy, precision, specificity, limit of detection, limit of quantification, linearity, and range. And important to include in your timeline is that method qualification can take a bit of time. Average timelines are 6 to 12 weeks. So you want to build that into your considerations. And finally, I want to remind everyone to not forget that in order to file for your IND, you must submit at least 1 month of stability. For a messenger RNA program, you're going to want to consider long-term storage conditions as well as accelerated, and you're going to want to choose a panel of assays that are going to be stability indicating. Then you're going to choose time points, at minimum out to 12 months. Oftentimes we encourage our clients to go to 24 or 36 months, depending on the length of their trial. But we do offer a variety of options, and we're here to happy to discuss your program to find the best fit for you. So I will end there and thank everyone for their attention.
Jessica and Anton will show yourselves and unmute yourselves. So we are very fortunate to have both of these guys. They both say Anton, one is not. One is actually Jessica. So that just shows you how you can put one person at two different places at the same time. So we're ready to do it. You've got one question already, and we want you guys to ask any questions that you have. And like I said, you can raise your hands if you want to ask your question live, or you can put it into the Q&A, and then we can see it. So the first question that we were asked is, what's the structural difference between cap 1 and clean cap?
I guess I can take that one. So
you're muted still. Sorry, Anton, we're not hearing you.
Anton, I think now you're muted. I was able to hear you a minute ago.
Can you hear me now?
Jessica.
Oh.
I can hear you.
Yes?
Not sure why.
Okay, so I can't hear you guys, but so there's a whole possibility of what you can do with CleanCap. You can have the correct Cap1 structure that you would find endogenously in nature, or you can also have unnatural caps. And so CleanCap offers the option of having either the natural Cap1 structure or whatever chemistry you want at the end of the 5' end.
Why we still have you guys here. And I was saying you guys just participated in our RNA symposium that we had in July, that we might want to just talk a little bit about some of the things that you guys, that you were asked at that time. And so I was, I think maybe you guys can just kind of add to anything you can kind of think of. And one of the questions I know that was big on everyone's mind is, how with COVID-19 and all the things that are going on, and just you give examples of how you had to accelerate, and being able to be really responsive. What are some of the other things that you think are impacting the industry? Are you're seeing more people outside of just COVID, outside of vaccines, becoming more engaged with mRNA, and looking at pursuing your services and asking you more questions about what you do?
You want to take that one, Jessica?
Sure, happy to jump in and answer that. I would say that absolutely, with the recent success of the vaccines demonstrated by Pfizer, BioNTech, and today some of Moderna's published data, I think that there's a lot of people interested in messenger RNA and the applications as it can be applied to vaccines. So there's definitely been increased interest in the vaccine space for messenger RNAs. Prior to this year, many people were using messenger RNAs for protein replacement therapies and especially for ex vivo gene editing applications, but vaccines has had a big insurgence in the last few months now. So it's good to see the success of these vaccines, and hopefully looking forward to everyone's lives going back to normal.
We don't have any
Jane,
more
I
questions.
can't hear you.
Yeah, okay. We don't have any more questions. Wait, something just popped up. Hold on, now they're coming. I was trying to give everybody a little bit of time. Here we go. In case of self-amplifying mRNA manufacture, any specificity should be attention to when comparing to other general mRNA manufacturing?
With self-amplifying messenger RNAs, one of the key parameters is that they're extremely long to contain the structural proteins for the replication. And so most of these constructs are 8,000 to 14,000 bases in length. And one of the things that's most challenging is keeping that full-length product after transcription. T7 polymerase is highly processive, so you do get a lot of full-length transcript from transcription, but then the purification process and additional enzymatic treatments that take place after transcription will begin to create fragmented sequences. You'll get physical shearing of the molecule. So one of the things that we recommend is having the shortest and fastest manufacturing process. A one-pot transcription and a one-pot purification is going to get you the most full-length product when it comes to a self-replicating construct.
Great. Another question that just came in. What kind of synthetic CleanCap chemistries are used for different applications? Why would you want one chemistry over another?
Anton, you want to answer that one?
Sure. So I think that one of the opportunities of CleanCap is that you can change the sequence of the 5' end. Our original CleanCap molecule that we mostly employed started with AG. However, we noticed that a whole bunch of different viruses, alpha viruses, flaviviruses, all start with an A, actually AU. You could also imagine that you could do other things to enhance the binding of cap binding protein, for example, to the 5' end, or reduce degradation. And so CleanCap offers the option to be able to explore other chemistries at the 5' end that you couldn't access with, a polymerase start with an NTP. And so we haven't explored a huge amount of modified ends, but that is definitely a possibility that can be accessed through the use of CleanCap. You could also imagine that you could put a label or you could put a handle to be able to attach the mRNA to a substrate. So there's really a whole lot of different, different applications.
Okay. Your next question is, will having a PD phase between phase 1 and phase 2 pose a risk in clinical data in terms of efficacy for the client?
I mean, there's always a risk if you're changing the manufacturing process, but one of the considerations you have to make is that small scale manufacturing for phase 1 may not directly translate to a commercial manufacturing scale. We absolutely advise our clients that on different pathways to choose the best manufacturing method for their product, and we have methods that are directly scalable. But depending on time considerations and amount of material needed for a phase 1, you may choose to do a small scale manufacturing process and then do a process development bridging study before going to phase 2 and later. So I would anticipate that if you're going to do a process development phase between phase 1 and phase 2, you'll probably need to generate some new toxicology data to demonstrate that the product is still safe and effective in an animal study.
Great, thank you. Is it possible to design a self-amplifying CRISPR-Cas9 mRNA, and what would be the drawbacks of this technology?
I mean, you could certainly express Cas9 from a self-amplifying RNA. So I guess one of the drawbacks is that the self-amplifying RNA is recognized as being something foreign within the cell. And so in general, what we think is that for a genome editing application, you want to be as clean as possible and not induce innate immune sensors such as PKR or MDA5, RIG-I, that those would be disadvantageous in a genome editing application. Whereas in the context of a vaccine, having the double-stranded RNA there is it can be considered to be advantageous and actually part of the drug. So the way that immunity works is that it requires more than one signal in order to prevent autoimmune disease. And so when an antigen is presented in the context of a danger signal, which is something that's the cell recognizes as not being quite right, that makes for a more potent immune reaction. You can kind of think of it like two-part authentication with your bank. You want two signals that let you know that there's something foreign there. And so in that case, the self-amplifying nature of the RNA and the fact that it looks like a virus to the cell is actually advantageous for the application. And can actually lead to apoptosis of the cell eventually. In the context of genome editing, you generally want to preserve that cell. And so, while you could express Cas9 from that kind of context, I don't know that you would necessarily want to.
Great. Okay, this is our last question, and it was, does vaccine capping enzyme generate CAP-O or CAP1?
I can take that one. So vaccinia virus capping enzyme generates a CAP0 structure. There's a second enzyme that can be employed called 2'-O-methyltransferase. And this, in a second enzymatic step, adds a 2'-O-methyl group to the sugar to create a CAP1 structure. But it requires both of those enzymes. And these enzymes are kind of finicky and difficult to produce. They also require a methyl donor, which is S-adenosyl methionine, or SAM, which is temperature sensitive. And so we found when we were doing a lot of enzymatic capping that you could have variability at these different steps. So in some cases, we would see partially methylated RNAs that lacked, say, the 7-methyl-G, that methyl group on the G, or lacked the 2'-O-methyl group on the first sugar. And so you can achieve a CAP1 structure, but it requires the use of both the vaccinia virus capping enzyme and the second enzyme, the 2'-O-methyltransferase. One of the things we like about CleanCap is that we're removing two enzymes, and those are some of the most finicky parts of the reaction. And so those enzymes have to be qualified. And so one of the nice things about CleanCap is instead of doing enzymology, we're basically just doing chemistry.
This is, I think, a follow-up to that question, but in that case, the vaccina-capped RNAs wouldn't yield any immunogenic data if injected into mice.
I mean, people have used vaccinia virus enzymes and the 2'-methyltransferase in order to use self-amplifying RNAs. I'm not sure what would happen if you omitted the 2'-methyltransferase.
Well, thank you both, and thank everyone for attending today. This was outstanding, and we really appreciate it. And I think it gave us also, oh, sorry, somebody asked, okay, let's see if this one more question came in real quick. Oh, it was just a thank you. So thank you guys. Thank you both for joining us today, and thank everyone else for attending and your good questions. And remember, this will all go on to an on-demand on the site and will be available for the next 30 days. Thank you, guys.
Thank you.